We're going to begin our discussion today with where we left off yesterday. So if you remember correctly from yesterday's lesson, we ended up with metaphase, which was number 3 on our diagram. Now, what can you tell me about metaphase? Okay, what do we associate with metaphase? Meeting in the middle. Okay, the chromosomes are attached to the spindle fibers, and they are aligned right here in the middle that we called the equator. Good. Okay, so if you remember, what was the little device that I told you, the little memorization helper with the stages? PMAT. So what's coming next? A, which is anaphase. So here is anaphase coming right at you. Okay. The chromosome pairs start to separate and move to opposite ends of the cell. So what we have happening is the chromosomes were lined up in the middle and they look like X's, correct? What we're going to see here is that those X's are starting to break apart, if you will. So where we had the centromere, and where they were lined up on the equator, they are being almost like pulled to each end of the cell. So here is the diagram that you need to draw in your box number 4. Okay, so now we can see that the chromosome pairs have separated and they are moving to opposite ends of the cells. Okay. After anaphase comes telophase or telophase, which is number 5 on the diagram. 2 new nuclei form. The chromosomes change to chromatin, which are threads rather than the rods that you saw before. So the chromosomes change to what we call chromatin. And finally, mitosis ends. So we can say that this whole process of mitosis duplicates or synthesizes the genetic material and then separates it into 2 new nuclei. Here is the picture that you need to color in on number 5, and then we'll talk about it once you get that colored in. Okay, as we are looking at this, we can see that certain things are going on here, kind of like the reverse of earlier. What are these little dashes, do you think? Where did we see those little dashes like that before? Okay, check out your diagram. Where did we see those before? Fletcher? In prophase. And what happened there? The envelope was disintegrating, right? So the nuclear membrane, or the nuclear envelope, was disintegrating. Now we can see that it is starting to reform. So I can see that I have two nuclei starting to form there. This, folks, to me, would be an earlier telophase. If we would get into a later telophase, we would see that that envelope was actually, that nuclear membrane was actually present then. So you could tell, oh, this is kind of early stage and a later stage. Okay? Now, as I get to our final stage, cytokinesis. Okay, cytokinesis is where the cytoplasm separates and two new cells are formed. The cytoplasm separates, two new cells are formed. Now, it is different in plants than it is in animals. If we look at the plant cell, the cell wall forms down the center. So, right here is what we call the cell plate. So, during telophase, this cell plate starts to form, and then in cytokinesis, the cytoplasm is separated, and now I have two new animal cells, and right here are my two nuclei. Now, for the plant cell up here, it's a little bit different. See how the cell starts to pinch right here. The cell starts to pinch right here, and we actually call that the cell is cleaving. So that is cleavage there. Once that happens, the cell splits into two different cells, and you can see that the nuclear membrane is almost reformed here. So we almost have a new nuclear membrane and two cells, and we refer to these two cells as daughter cells. Daughter cells. Now, let's put it all together. Okay? Let's put it all together. Cells multiply by dividing. Kind of funky, huh? Not quite the multiplication and the division we're thinking about, is it? Okay? But in our bodies, our cells were multiplying by dividing. Kind of interesting. Results of mitosis. The results of mitosis are that we have 46 chromosomes in every cell in your body. They're paired up, so we have 23 pairs. Now, you're going to see later on that sex cells only have half the number of chromosomes as which a body cell has. So we'll learn that later on. You know, our body cells have 46 chromosomes. The sex cells. We're talking about the sperm. We're talking about the egg. The sperm and the egg only have 23. Why do they only have 23? What ends up happening? They combine, and when they combine, we have 46. Okay? So that's why those sex cells only have 23, due to the combination, and gives us a little bit of genetic variability. Now, body cells. All other body cells have 46 chromosomes. Now, what we're going to look at right here, this is a karyotype. Okay, I don't know if we'll talk about karyotypes or not, but as you can see, these are karyotypes where we have the homologous pairs right there. So you can see these have been duplicated, and these are the sister chromatid. Now, I would say these are probably somewhere, I think when they're doing karyotypes, might be somewhere around metaphase, because you remember that the sister chromatids lined up at metaphase. But nonetheless, you can see that these are sister chromatids. Now, if you would count these, there are 22 pair, and here's the 23rd. You probably can't see it, but this right here says that this 23rd, the sex chromosomes, this is an X and a Y. The X is the bigger one here, and the Y is the smaller one. So if you know anything about genetics, what's XY? A male. So this would be the karyotype of a male. And it has 22 pair of autosomes, what we call them. And then here's the 23rd, the sex chromosomes that are not homologous. Now, if you look on the karyotype, how are they grouped? They're grouped by ones that are similar, but as I go down, as my numbers are getting bigger, what's happening to the chromosomes? They're getting smaller and smaller and smaller. So we'll deal more later on about these karyotypes. I just wanted to show you the 23 different pairs of chromosomes that we're dealing with. Okay?